All right, let's talk about real-world evidence. And we've already seen examples of real-world evidence in the past couple of days. So this some of this will be a little bit of a review. We don't want to spend too much time on it, but I do need to level set. All right, we'll go old school. Okay, so if you want to do any work with the Real-World Evidence Core, this is the team, myself, Anita Mendoza at Cincinnati Children's, Colin Rogerson at Indiana, and then Sean Granis, who is around here somewhere, I'm sure. Oh, there he is in the back. Sean Granis at Regent Street. And everybody here has multiple affiliations. I think Anita's worked at all these places, so we're very familiar with each other's hospital systems and things like that. Okay. So in the past 5 years, there's been some large trends in real-world evidence that I think impinge on us quite a bit, not the least of which is we started in 2021, and that was a very forward-thinking initiative because we were a small optional core in the grant, and the goal was we need to develop ways to just get everybody talking and sharing data and sort of make a resource available. And that was the first time that was going to be done. But in the larger world, the EU started the Darwin Consortium, which uses OMOP data. It's a common data model for sharing EHR data across hospital systems. They started this consortium to real-world evidence at scale within the EU. The FDA came out with a guidance on using real-world evidence, and then came out with another guidance that was much more specific. We're not yet at the ICH E9R1 technical manual of how to analyze clinical trials yet in this world, but these two documents, especially the 2024 one, are getting us way closer to that than we had before. These are guidances that, even though real-world evidence was allowable by the FDA since around 2016, this guidance helps formalize things to the point where organizations are much more comfortable operating in this space and really encouraging PIs to work in a rigorous way. And so these are fantastic documents that have come out in the past few years. And I'm happy to say that our core has matured assets in this space to the point where we're ready to scale a lot, and we'll be talking about that today. So what's the role of real-world evidence? In a phrase, it's just post-approval learning, right? So comparative safety and effectiveness for medications that are in routine use, so not experimental therapeutics as such. But we have a lot in the clinical trial space. Generalization is an issue. Why does a pragmatic clinical trial even exist? Because patients are typically on multiple drugs and have multiple diseases. Rare or delayed harms, very hard to do that in a clinical trial, the kind of space that you have. We want to know, how's the kid at 5, 7, 8 years? That's a much tougher thing to do. Real-world evidence sort of naturally works well in that space. And then there's other sorts of questions that you can ask with this that are just harder to do in other contexts. And not the least of which is underrepresented patients and populations like preg-lac and pediatrics, for example. And so real-world evidence has a role to play in this space, and we've seen some good examples of that. Here's 3 basic types of real-world evidence: pharmacovigilance, pharmacosurveillance. We actually saw a number of individuals from Dr. Longley's department doing good work in that. A lot of that can be automated. It's fantastic for signal detection, finding things that are unexpected. Pharmacoepidemiology, we saw some wonderful examples of that with SSRIs yesterday. That's asking these broad population-based questions, sometimes at a population, sometimes at a cohort, understanding medication use and outcomes. And there's this target trial emulation, which we did hear the groundwork for one being done yesterday. And so that is a trial-like causal inference that is designed to answer a practical clinical question. So should you start A or B at recognition of pregnancy? Or I guess the more relevant question is, should you stop drug A or B at the recognition of pregnancy? And so what is a clinician going to do? These are often yes/no questions, but the goal of target trial emulation is to reduce bias as much as possible and come up with an answer that is clinically actionable. And so target trial emulations are an important thing that's been going on in this field. It's one of the macro trends that we've identified in real-world evidence across the past 5 years, especially, and how mature a lot of this has come. One area is data linkage. And so we're going to talk about some various algorithms that we've defined to help data linkages. But taking a data set and trying to get as much adjunct information as possible A lot of that comes with data sharing is a big part of this. There was an EO in this past year saying the federal government needs to make data more available to people. In response to that, we saw all of us and N3C and other large NIH consortia redoing their data usage agreements in ways that my regulatory people were quite excited about, so much more favorable for the end users in terms of getting access to data and sharing it. And so this is becoming more of a zeitgeist. Yesterday, Lars Bodie talked about a contract between San Diego and University of Washington that happened in 48 hours. The idea of a master service agreement is something that is very old in legal parlance as a term of art, but now we're seeing a lot more universities and a lot more companies saying, yeah, let's just do that because we'd rather not renegotiate everything from scratch. The lawyers are costing us a lot of money and not necessarily adding as much safety and value as they might ought to. So that zeitgeist of let's link data, let's share data is definitely maturing in the past 5 years. We're seeing a lot of positive developments. I mean, when you're at the bottom, you can only go up, so I have every reason to be optimistic. It can only get better from here. I touched on target trial emulations. I'd say causal designs have definitely matured, especially in the past 5 years. You know, we all know that when ChatGPT came out in 2021, there was this inflection point on the use of AI in academics and in research. The same thing is happening in the causal modeling framework. We went from one computer scientist who won a Turing Award writing books on it to now the elbow, the inflection is coming where we're seeing more and more target trial emulations. We're seeing more and more causal modeling of all sorts and also in the pharmacoepidemiology space. And so these things are out there, and so we're riding right on that wave. And then we're seeing the industrialization of evidence in terms that goes hand in hand with data sharing. We're seeing things like the knowledge base, and we're seeing all these other projects that are just putting the data freely up on the web so that anyone can query it and access it in more and more an automated, metadata-driven, AI-ready type fashion. And so these trends are all incredibly helpful for what's going on in the real-world evidence space and where we're capable of going now. So let's get a little specific about what we're doing in the core. The first thing you have to do is you need to identify pregnancy in these large databases. And so we've developed an algorithm for that. We had about 1 million known pregnancies. You have to have a gold standard data set to operate on. And you can see that the positive predictive value was 92%, negative predictive value is 85%. So highly specific model. There's a little bit of sensitivity. Isn't quite as high as everyone always wants, but in a large database like this, you still get a very large sample size that's mostly correct out of this kind of work. And so very nice, robust algorithm. This is a deterministic algorithm, which, you know, AI is the sexy thing, but a deterministic algorithm is easy to interpret. If it starts to break or goes wrong, you can just open up the hood and fix it. You know, COVID happens or something macro occurs, you can easily make adjustments to it. Very cheap to run, very fast. And so there's a lot of benefits to that. If you can't use AI, then you definitely want to operate in this space if at all possible. So that's what happened there. So we can identify pregnancies very reliably. Lactation is going to take a minute to unpack. So we need to know if we want to do real-world evidence studies on breastfeeding and exposure to drugs and lactation, we need to be able to identify that. That is not routinely captured in structured data in the electronic health record. And so the solution there is to read the clinical notes. So this is where AI comes back in. And so we started a study with Ohio State University and Nationwide Children. So we had moms' notes from the nursery and right after birth. We've also got the kids' notes all the way through the first year of life. Say, can we go ahead and figure out a way to figure out, was there positive evidence of breastfeeding in a note? Is there negative evidence of breastfeeding in a note, meaning it stopped or wasn't occurring? Or was it agnostic? And most notes are on the agnostic side. But if you can classify every single note that way, you can build up a sparse data structure that tells you roughly when the kid started and stopped breastfeeding. And so the model on the infant side, actually a deterministic model, turns out to be very lightweight and very workable. On the mom side, an AI model can do incredibly well with an F1 of 96%. And so we went ahead and applied this to all 55,000 linked moms and babies between OSU and NCH, just so we could turn the crank and learn more about this problem. There's some complexity here, not the least of which is this is what the data structure looks like when you finish this. So we have lots of kids, and everybody sees lactation consultant almost universally in the nursery, and so you see lots of blue dots on the left, and then as time goes by, we have A, less data, and B, you're going to see evidence that breastfeeding has stopped, but you're not going to get a precise date as to when it stopped. That is just not in the notes, right? And so we have this data structure that is sparse, unevenly sampled, and the ends are very fuzzy. And so statistically, this is a whole new area because we did a let search to see, well, what is the intelligent stuff to do with a data structure like this? And there's not a whole heck of a lot out there. And so we're definitely moving into a new domain with this. There's going to be some interesting papers that can come out of this. How do we analyze this data effectively to account for the complexity, which has some uniqueness here? And then, again, this was just the beginning. We want to roll this out to the entire core's data. But now we have an opportunity to go back. This is the same slide above. I just added some stuff at the bottom. Now we have an opportunity to capture a little bit more about the complexity that was in there. And so yesterday there was a mention of goat milk being used. We didn't capture that in this first go around. I'm not sure how common that is. I did talk to a colleague who tested breast milk years ago, and she said 10% of the samples were adulterated with either soy milk or cow milk. So I don't know if that's ever going to be in the notes. There's just a lot of information that isn't there, but we're going to push as far as we can. And then the thing that always kind of I'm curious about is the concept of dosing even possible with the information that we have. I don't know if there's something intelligent that we can do in that space with maybe lots of uncertainty that we can manage. But it seems like a place that's interesting to talk about, at least. I don't think it'll ever be perfect, but can we do something better than nothing, which is what we can offer at the moment. So to be continued, a very active area of development, a very important area. And then we have to find moms and babies. We have to link them together. And we have an algorithm that works incredibly well based off of training from 10 million mom-baby linkages from over 120 hospitals. And so this is a large data set, very robust algorithm. The F1 score is 92%. The precision is 98%. So we can do a really good job. I will say a fair number of mom-baby linkages are going to be deterministic. You don't use the AI algorithm for everything because if there's a pregnancy fact table and there's you can just do the direct linkage in the medical record. So whenever you can, you don't use the AI algorithm. You do it deterministically, and so you're going to find a lot of this is deterministic. But when you can't do that because of reasons like a fair number of babies leave the birthing hospital with the name baby, and then when they come to the children's hospital, they have a name. And so there's some times when you can't do deterministic linkage. And so that's where the AI algorithm comes in. So you don't always have to do this, but when you do, we have a pretty reliable algorithm. And so that puts all the basic pieces together to have a more mature Real-World Evidence Core data asset that's available. And so here's what we have. We have all of our data mapped to OMOP, and as I mentioned, this is a common data model that's used even to trade data with Europe. I mean, we can very highly portable. And if folks aren't in our particular consortium, but they have OMOP mapped data, which is actually pretty common in academic systems, they can still come to us and join that data with what we're already doing. And so this is a very portable and expandable framework. Depending on what you're doing, you're going to have somewhere between 1 million to 10 million mom-baby dyads available to do certain types of research. That is a big sample size that's necessary for reasons we'll go over in a second. All the data has been geocoded, so we can link these data to area level indicators, and we're always looking out for ways to sort of sharpen that up and improve our health-related social needs data. And then the next step, the thing that we want to finish as soon as possible, is the lactation phenotype. We want to validate that outside of NCH and OSU. We want to roll that out across the entire sample so that this is available. And then our colleagues at UCSD can do pharmacoepidemiology and some other things on a larger American data set. Nothing against the Danish data set. It's a poor model for the complexity of the American healthcare system, but it's a fantastic data set otherwise. But here, we can give you American data on that scale. So the large sample sizes are necessary. I mean, if you're going to detect rare events and if you want to improve matching for causal modeling, you can only do that if you have a lot of opportunities to get a good propensity score match or some of the other causal modeling techniques. And so this is vital for moving forward in this space. And the thing that, again, we rolled up our sleeves and did the stuff that wasn't sexy. The least sexy thing that we've done is governance. So getting an IRB, a single IRB across all the institutions, is something where we have Indiana 100%, and we have to change one word on the protocol, and we'll have Cincinnati on 100%. along with OSU at Regent Street and Nationwide Children's. So the SIRB is a really big deal. It took us a long time to get that over the finish line. But that's the framework that allows us to sort of share data and hang out that signal and say, hey, let's all work together. And so I would say, based on the stuff that we heard in the last couple of days, come and talk to us about surveillance and epidemiology and target trial emulations, because we've got some interesting things to offer you. And so I will end it there. I guess people need to come up to the stage now.